Hi, my name is Kelly Kim, and I am a PR manager at Olix Pharmaceuticals. Today, I'm here to briefly talk about our company. Olix Pharmaceuticals is a biotech that develops novel therapeutics based on proprietary RNAi technology. We are based in Suwon, South Korea, with about 74 employees, and 53 of them are concentrating in R&D. Our company was founded by Dong-gi Lee in 2010, and we got listed on KOSDAQ in 2018. Our overseas subsidiaries are located in Boston and San Diego. Developing new therapeutics requires experts from various fields. Olix comprises in-house expertise covering from R&D to commercialization, along with an excellent scientific advisory board members. So let me move on to talk about our core technology. So what exactly is RNA interference? As you know, DNA makes RNA makes protein. And if the produced protein makes a disease or it has a problem, it can cause a disease. And the first and second generation drugs inhibit the activities of these produced proteins. However, our generation, the third generation oligonucleotide drugs, inhibit protein synthesis itself by cleaving the mRNA using oligonucleotides such as siRNA. As you can see, 85% of the disease-causing proteins are not targetable by first and second generation drugs. And it takes about 3 to 5 years to identify potential drug candidates. However, with our third generation drug technology, it becomes possible to theoretically silence all disease-causing genes. That is why the RNAi technology is the most powerful oligonucleotide technology. Through its catalytic process, it becomes possible to effectively cleave mRNA and silence the disease-causing genes. But there are some limitations of existing RNAi technology. First, side effects. Second, delivery issues. So in order to overcome these limitations, Olix has successfully developed an asymmetric siRNA platform technology. As you can see in the red box, we have different numbers of oligonucleotides in sense and antisense strands. And this reduces the off-target effects as well as other side effects. Patent is granted for this structure in Korea and other countries. Furthermore, we have developed cell-penetrating asymmetric siRNA platform to overcome the delivery issues. By adding the lipophilic moiety, we have successfully delivered the structure all the way to cytoplasm by penetrating the cell membrane. Right now, in RNA market, a lot of companies are targeting the liver diseases. Olix has also secured asymmetric GalNAc platform for liver-targeted delivery and are actively finding targets for liver-related diseases. So, how is Olix's platform technology so exceptional? In order to select the potential drug substance, we can simply change the sequence in antisense strand, which reduces the selection time for drug candidates drastically from 3 years to 3 months. With this time allowance, multiple pipelines can be expanded with reduced cost, and flexible business strategies became possible, such as early stage licensing. So here is our pipeline overview ranging from skin, eye to liver. I'm going to be talking about our core programs. First, on hypertrophic scar, Olix 101A. Hypertrophic scar and keloid scars are skin abnormalities caused by injury and surgery. And right now, there are no current treatments in the market. So Olix decided to target CTGF, a major protein that promotes the fibrosis in our cells. And we have verified the effective suppression in animal models. The pipeline is right now in phase 2 clinical trial in Korea, and in the US, IND is getting filed very soon. Next up is our ophthalmic program, Olix 301A and 301D, related to age-related macular degeneration. There are two types of AMD, wet and dry. And there is a drug developed for wet AMD, but for dry AMD and subretinal fibrosis, which is caused by wet AMD, there are no drugs available at the market. So we are developing the first in-class drug for both wet and dry AMD simultaneously, and verify the efficacy in the mouse and NHP models. For our eye program, an ophthalmic company located in France called THEA showed high interest, and we have signed the out-licensing agreement with them back in March 2019. The total volume was about 63 million euros, patent covering from EU, MEA to Africa. We're also developing a drug for both subretinal fibrosis and wet AMD and verified the efficacy as well using the same protein. Next, for our pulmonary area, we are targeting idiopathic pulmonary fibrosis, which is a fatal disease that leads to death within 3 to 5 years after diagnosis. With IPF, we have successfully suppressed the target gene expression, and we've seen effective suppression of fibrosis as well. And the research was done with the agency in Singapore. Up next is the hot new issue, COVID-19. With using our CPhI RNAi platform technology, we have successfully identified the lead candidates and verified the efficacy in cells and assays. So now we are moving on to animal efficacy testing, and we are waiting for the results to come out. Last but not least, for liver diseases, with our own proprietary GalNAc platform technology, we are actively discovering the lead compounds for targets suggested by our liver experts at Harvard. And we have recently signed the R&D supply contract with a biotech company in Europe. We're currently testing and developing lead candidates for 4 different liver targets that are requested upon the party. And if we do identify the targets, we are going to move on to actually licensing out the targets. So that is all for our company summary. I'd like to thank you very much for listening to my speech. Thank you.